Synapses are the place where one neuron connects with another neuron or a muscle cell. Here, impulses, so-called action potentials, can be transmitted. In the synaptic terminal, membrane-enclosed vesicles, which are called synaptic vesicles, can be found. Those contain tiny amounts of transmitting substances, which are called neurotransmitters. If the action potential reaches the synaptic terminal of a neuron, those vesicles are triggered to migrate to the presynaptic membrane, with which they fuse and thus release the neurotransmitter into the synaptic cleft. They bind onto receptor molecules located at the postsynaptic membrane and cause Na+ channels to open. Sodium ions therefore enter postsynaptic cell and depolarize it. The more ion channels are opened, the greater is the resulting depolarization, and an action potential is triggered to be transmitted within the central nervous system. This is the process of an excitatory postsynaptic potential, so-called EPSPs. Besides the excitatory synapses, inhibitory synapses appear to the same ratio in the central nervous system. Excitatory synapses lead to a depolarization of the postsynaptic membrane, while inhibitory synapses cause an hyperpolarization. Here, another synaptic knob docks onto the presynaptic cell, which causes neurotransmitters to be released into the synaptic cleft. Those enable the chloride ion channels of the presynaptic cell to open, and thus chloride ions which are negatively charged enter the presynaptic cell and cause the vesicles to stop migrating to the presynaptic membrane. This is why less and less neurotransmitter are released into the synaptic cleft, and thus more and more Na+ channels at the postsynaptic membrane are closed. This is the reason for the postsynaptic membrane to remain below the threshold, and thus no action potential is triggered. This inhibition is important for the regulation of muscle movement. Several toxins can affect the synapse in different ways. Toxins either act at the presynaptic knob, in the synaptic cleft, or the postsynaptic membrane. The impact is explained with the following examples. Firstly, curare. Curare is a mixture of various plant toxins, such as the borg from liana, that is used mainly by natives in Southern America. If the toxin passes into the bloodstream of prey, the skeletal muscles that we voluntarily use, are paralyzed. It binds on the receptor of the channels, leaving them blocked, so the Na+ ions can only pass through at little amount. Thus, only a few Na+ ions can diffuse into the postsynaptic cell. So when an action potential is trying to be submitted, the transmitter are released into the synaptic cleft. On many of the receptors, curare is already attached, enabling the transmitter to bind and the Na+ channels to open. This prevents a postsynaptic potential to develop, so the transmission of an action potential to the following synapses is not possible anymore. The chain of events causes an arising of muscle paralysis, which causes the lungs to collapse. Secondly, alpha-latroxin. One bite of the black widow has the following impact on the presynaptic membrane. Normally, due to the IPSP, calcium channels close after an action potential is submitted, but under the influence of alpha-latroxin, the channels are permanently open, which leads to many transmitters diffusing into the synaptic cleft, causing a permanently influx from Na+ into the postsynaptic cell. This means that there are steady action potentials triggered without any stimuli. The spider managed to paralyze the muscles with its poison, causing chills, pain, shortness of breath, and in the worst case, death, due to respiratory paralysis. Thirdly and lastly, botulinum toxin. The bacterium Clostridium botulinum lives under these anaerobic conditions like contaminated tins. Botulinum toxin prevents the release of acetylcholine into the synaptic cleft. It degrades a protein that is present in the membrane of the synaptic vesicles, inhibiting the vesicles which contains the neurotransmitter acetylcholine to fuse with the presynaptic membrane. So, action potentials can no longer be transmitted from the nerve to the muscle, causing muscle paralysis and under high doses leading to death.
Benji!